Introduction. Pseudoexfoliation syndrome, X, is an age-related eye condition characterized by the accumulation of flaky, white fibrillar material on structures in the front part of the eye, such as the lens capsule and pupillary border. This material is rich in elastic microfibrils and other extracellular matrix proteins, so PX is often described as an elastosis, essentially an overproduction of elastic fiber components in the eye. Over time, PEX can cause elevated eye pressure and trigger a form of glaucoma called pseudoexfoliation glaucoma that damages the optic nerve and can lead to vision loss if untreated. Patients with PEX also appear to have higher rates of vascular diseases, for instance, stroke or heart disease, suggesting systemic factors may be involved. Scientists have noted that patients with PEX glaucoma often have higher blood levels of the amino acid homocysteine than people without the disease. Homocysteine is a byproduct of normal protein metabolism. It comes from the essential amino acid methionine. Diets very high in protein, especially animal protein, can deliver a lot of methionine. If the body cannot fully convert homocysteine back into other useful compounds, homocysteine can accumulate in the blood. In this article, we explore how high-protein diets and one-carbon metabolism, which depends on B vitamins like folate and B12, might influence homocysteine levels and thus potentially affect the risk of developing pseudoexfoliation glaucoma. We will also discuss how abnormal homocysteine might disrupt enzymes involved in building and remodeling the eye's connective tissue, notably LOXL1, a lysyl oxidase enzyme that cross-links elastin fibers. Finally, we suggest how future studies could be designed to test these links using detailed dietary data, genetic testing, blood biomarkers, and advanced eye imaging. Protein intake, methionine, and homocysteine. When you eat protein, your body breaks it down into amino acids, the building blocks of proteins. One amino acid, methionine, is found abundantly in many proteins, especially in red meat, eggs, and dairy. Methionine is converted in the body to homocysteine. Normally, homocysteine is then either recycled back into methionine or converted into cysteine, and this process depends heavily on B vitamins: folate, vitamin B9, vitamin B12, and vitamin B6. If these vitamins are insufficient, or if dietary methionine is very high, blood homocysteine levels can rise. Controlled diet studies in healthy volunteers show exactly this relationship. An 8-day high-protein diet, about 21% of energy from protein versus only 9% in a low-protein diet, led to significantly higher post-meal homocysteine levels throughout the day, even though fasting homocysteine didn't change much. In other words, after people ate protein-rich meals, their plasma homocysteine spiked higher than it did when they ate low-protein meals. The researchers noted that a high protein intake, and hence a high intake of methionine, the sole dietary precursor of homocysteine, may raise plasma TAC concentrations. In practical terms, this means diets very rich in meat, fish, eggs, or other high methionine foods can transiently increase homocysteine unless balanced by enough folate and B vitamins. It is important to emphasize the role of B vitamins. Even people who eat a lot of protein may keep homocysteine under control if their diet supplies plenty of folate, B12, and B6. Conversely, some people on vegetarian or vegan diets, who might have lower methionine intake, actually have higher homocysteine if they are deficient in vitamin B12. For example, one review showed that vegetarians, who often do not get meat-based B12, had higher average homocysteine levels than omnivores, 13.2 versus 10.2 mg, largely due to B12 deficiency. This illustrates that it's not just protein per se, but the balance of nutrients. Without enough vitamin B12 and folate B6, homocysteine rises in many different diets. Pseudoexfoliation syndrome and homocysteine levels. Several clinical studies have now examined homocysteine in patients with pseudoexfoliation. They consistently find that people with PX, and especially those who have progressed to glaucoma, tend to have higher homocysteine. For example, a prospective study compared 30 patients with PEX glaucoma to age-matched controls. The PEX glaucoma group had an average plasma homocysteine of about 16.8 mmHg, whereas the controls averaged 12.4 mmHg. Even more striking, 50% of the PEX glaucoma patients had homocysteine above 15 mmHg, a common cutoff for hyperhomocysteinemia, while only 10% of the controls did. Similarly, another study found that both PEX syndrome and PEX glaucoma patients had significantly elevated plasma homocysteine compared to normals, but patients with ordinary, primary open-angle glaucoma did not. In short, pseudoexfoliation appears specifically linked with high homocysteine in the blood. A 2012 meta-analysis pooled many studies and confirmed this pattern. Across 485 PEX glaucoma cases and 456 controls, the average homocysteine was about 3.4 millimoles higher in the PEX group. The PEX glaucoma patients also had slightly lower folic acid levels than controls, though their B6 and B12 levels were similar. Importantly, the meta-analysis found no clear association between the common MTHFR C677T gene mutation and PEX glaucoma risk. This suggests that while homocysteine levels are higher in PEX, simple MTHFR genetics alone does not explain the risk. MTHFR is one of the key enzymes that helps process folate and homocysteine. Nonetheless, the combination of a high methionine diet and marginal B vitamin intake could exacerbate homocysteine buildup, especially in genetically susceptible individuals. Taken together, these findings raise the hypothesis that dietary methionine and homocysteine may contribute to the development or progression of PEX. If high-protein diets chronically elevate homocysteine, this could affect the eye tissues. Indeed, PEX patients often show not only these biochemical changes, but also changes in their connective tissues, such as weakened zonule fibers holding the lens, altered iris, etc., that might be sensitive to homocysteine's effects. Extracellular matrix, LOXL1, and 1-carbon metabolism. The material deposited in PEX is highly cross-linked and rich in elastic fiber components. It contains elastin microfibrils, including proteins like fibrillin, collagens, fibronectin, and other extracellular matrix (ECM) proteins. The genetic defect most strongly linked to PEX is in LOXL1, lysyl oxidase-like 1, an enzyme that normally helps cross-link elastin fibers. LOXL1 belongs to the loxidase family, copper-dependent enzymes that catalyze crosslinks in collagen and elastin by deaminating lysine residues. In fact, scholarly reviews note that LOXL1 seems to be specifically required for tropoelastin crosslinking and has been shown to be involved in elastic fiber formation, maintenance, and remodeling. In other words, LOXL1 is critical for healthy elastic fiber assembly. In PEX eyes, LOXL1 is both genetically and physically implicated. Certain LOXL1 gene variants dramatically increase PEX risk, and proteomic analyses have detected LOXL1 protein itself within the exfoliation deposits. For example, Shiwani Sharma and colleagues used mass spectrometry on surgically obtained PEX material and confirmed that peptides from LOXL1 were present in all samples tested. They also found proteins like apolipoprotein E, clusterin, complement C3 fibulin, and others. This indicates that LOXL1 is a substantive component of the abnormal fibrils. So why would homocysteine be important here? High homocysteine, or one of its reactive derivatives called homocysteine thiolactone, can chemically damage proteins like LOXL1. Biochemical studies show that homocysteine thiolactone is a strong irreversible inhibitor of lysyl oxidase activity. Specifically, homocysteine thiolactone can bind to the enzyme's active site and render it inactive. If this inhibition happens in the eye, it could impair normal cross-linking of collagen and elastin. Thus, excessive homocysteine might contribute to abnormal elastic fiber homeostasis and to the accumulation of incomplete fibrils that characterize PEX material. In addition, 1-carbon metabolism is intimately connected to the supply of molecules needed for ECM production. For example, 1-carbon pathways involving folate and B vitamins help generate glycine and other amino acids required for collagen synthesis, as well as S-adenosylmethionine, SAM, the universal methyl donor. Indeed, the metabolomic study found that S-adenosylmethionine levels were significantly lower in the aqueous humor of PX patients. Lower SAM can lead to global hypomethylation, potentially altering gene expression of extracellular matrix proteins or enzymes. Moreover, the metabolomics analysis specifically highlighted the cysteine and methionine metabolism pathway as one of the most disturbed in PEX eyes. This strongly suggests that changes in 1-carbon metabolism and homocysteine handling are linked to the disease process in pseudoexfoliation. In summary, there are plausible biological pathways connecting diet and 1-carbon metabolism PEX pathology, Methionine-rich diets raise homocysteine levels. Vitamin deficiencies, folate, B12, B6, or common MTHR variants can further elevate homocysteine. Elevated homocysteine and its toxic metabolites inhibit LOX-LOXL1 activity, potentially disrupting elastin cross-linking in the eye. PEC's tissue is composed of cross-linked elastic microfibrils, and LOXL1 function is known to be crucial for elastogenesis. Altogether, this suggests that if one carbon metabolism is off balance due to diet or vitamin status, the eye's connective tissues may accumulate abnormal fibrillar material. Proposed study design. To test these ideas, researchers could establish a prospective cohort study focused on dietary protein, homocysteine, and PEC's development. Adults age 60 plus without PECs at baseline would be enrolled. At the start, each participant would provide very detailed dietary information through food diaries or validated questionnaires to estimate total protein, methionine, and other amino acid intakes, along with intake of folate, vitamin B6, B12, etc. Blood samples would be collected to measure plasma homocysteine and levels of B vitamins. Participants would also be genotyped for key one-carbon metabolism variants, such as the MTHFR C677T polymorphism, and for the known LOXL1 risk alleles. Over time, for example 5 to 10 years, participants would undergo regular eye exams, including anterior segment imaging. Modern imaging methods, such as slit lamp photography, high-resolution anterior segment OCT, optical coherence tomography, or even confocal microscopy, can document early pseudoexfoliation deposits on the lens capsule, iris, and other structures. The key outcomes would be development of clinically evident PECs and PECs glaucoma, and quantitative measures of exfoliation material burden, for instance, grading the area of lens or pupillary deposits. By analyzing who develops PECs or PECs glaucoma, investigators could see if higher dietary methionine and plasma homocysteine, especially in people with low B vitamins or certain MTHFR genotypes, predict greater X risk. Such a cohort would clarify whether modifiable factors like diet and vitamin status influence PECs. If confirmed, this could suggest simple preventive strategies, for example, B vitamin supplementation or dietary adjustments, to lower homocysteine and potentially reduce PECs onset. Conclusion. Emerging evidence links high homocysteine with pseudoexfoliation glaucoma. Diets very rich in protein, high methionine, can raise homocysteine levels, especially when folate or B12 are inadequate. Meanwhile, homocysteine is known to interfere with lysyl oxidase enzymes that build elastic fibers in the eye. Because pseudoexfoliation is essentially pathologic elastogenesis in the anterior eye, a methionine-homocysteine imbalance could plausibly worsen or trigger the condition. In fact, blood tests show that many PECs patients have hyperhomocysteinemia and low folate. To fully understand these links, well-designed long-term studies are needed. We propose prospective cohorts that carefully measure amino acid intake, vitamin status, and genetics, and use detailed anterior segment imaging to track PECs deposits. Such research could reveal whether dietary interventions or vitamin supplementation might one day help prevent or slow pseudoexfoliation glaucoma. Sources: recent clinical and biochemical studies support these connections. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.